Hi guys, my name is Anggy Soto, and today I'm gonna talk to you about combining functional hairpin proof with disordered cleavage of CRISPR Cas12a protease to screen for B lymphocytic leukemia. So let's start. First of all, we have to know some concepts to understand what the authors did in this investigation. We have the hematological tumors that are a kind of malignant proliferations of this hematopoietic system. The B lymphocytic leukemia, or it could be known by the BO, is a kind of disease where our bone marrow produces too many kind of cells called lymphocytics. Their treatment of this disease is very aggressive. We have chemotherapy, drugs, and bone marrow transplantation. The incidence of this disease can be reduced if we screen the patients before present the symptoms of the disease. At the present, we use nucleic and detection technology that is widely used and can give valuable information because we have a gene called the gene PAX5A that is considered as biomarker of B lymphocytic leukemia because it's related with the differentiation and proliferations of the lymphocyte. Now we have the CRISPR that is an adaptive defense system guide by ribonucleic acid discovered in bacteria in 1978. The CRISPR-Cas system that is formed by two parts, the cRNA and the CRISPR that is associated to an enzyme called endonuclease, and it is a powerful technology used nowadays. The more widely used and major protein is Cas9, but in this project they decide to use Cas12 because it is more flexible and it doesn't require the participation of that cRNA, and it can also cut single-strand DNA non-specific. And functionalized hairpin proof is used to reduce false positives that they are used to detect target genes with a fluorophore. The electrochemical method used in this project was fluorescence spectroscopy. The objective of this project is combining functional hairpin proof with disordered cleavage of CRISPR Cas12a protease to screen the PAX5A gene related with B lymphocytic leukemia. We have two methods. The first one is the PACE, or is a kind of electrophoresis. There is a technique used to separate charged molecules by the migration in an electrical field, and the molecules separate according to their electrical charge. Our support medium can be used in the electrophoresis, but the one that we are going to use in these investigations is the polyacrylamide. It basically forms a kind of pores that are small to the other kind of gels, and they form a pore through which the protein passes. This method is used in the practice to group DNA fragments with their charge and the molecular's weight. The pores of polyacrylamide, as I said before, are very small, and the result is more specific than where we use another kind of gels. The other method that we have is the fluorescence spectrum detection of PAX5A. When we have the particles of the nitrogen bases and with a fluorometer, we are gonna know that the gene is there. Bueno, en cuanto a resultados, podemos ver una ilustración de lo que se analizó para detectar el sistema sensor, para reconocer el gen PAX5A, y también se busca reconocer la escisión desordenada del sistema CRISPR-Cas12. Inicialmente se toman unas muestras de los pacientes sanos y de pacientes enfermos. La horquilla funcional en este sistema lo que permitía era detectar de una u otra forma la presencia del gen. En pacientes sanos, la horquilla funcional no puede reconocer ese gen, ya que los primers o cebadores encargados de unirse a ese gen no lo hacen, no inician la reacción. La horquilla permanece igual y por ende no se crean los subproductos de la reacción. Después no hay interacción con las proteínas Cas12. Hay una proporción casi nula del gen en estos pacientes, como vemos en este diagrama, donde los pacientes que son sanos presentan una intensidad muy baja para dicho gen. Mientras que en los pacientes enfermos se hace efectiva la horquilla, se puede reconocer la frecuencia del gen y la secuencia con la que se hacen y volverse una secuencia de doble cadena, a la que en este caso si se van a pegar los primers y la proteína Cas12 reconoce la secuencia de ARN y se puede unir con el ADN marcado con el fluorómetro, y por eso se identifica la presencia del gen, mostrando que en estos pacientes que presentan la enfermedad sí la intensidad de la fluorescencia es muchísimo más alta para el gen que podemos ver. Los genes diana pueden distinguirse con éxito de los genes que no son diana, y es exitoso el sistema que se incrementó por esta investigación. En los otros resultados podemos ver en esta gráfica que cuando está el gen presente, El espectrófotómetro sí encuentra una fluorescencia y es mucho más fuerte, mientras que si el reactivo es leve, la intensidad de la frecuencia disminuye. En la imagen B podemos ver si el page fue efectivo con el fin de verificar si el sistema que se desarrolló es factible, para demostrar la capacidad de la horquilla funcional para abrir esos genes diana y que en realidad sirva para detectarlo. Podemos ver diferentes filas donde la electroforesis funciona. La banda A, la horquilla funcional del gen está normal, permanece igual debido a que no hay un gen que reconozca y no sufre un cambio conformacional, mientras que en la banda B se observa una interacción con el gen diana, que si solamente tiene el sedador, como en el caso de la banda C que podemos ver acá, la horquilla no va a cambiar su forma, así como en la banda A, concluyendo que solamente el gen objetivo, la horquilla funcional, sí es capaz de reconocerlo. Y por último, los resultados podemos ver la figura número 4, en la cual se demuestra que la fluorescencia sí está relacionada con las concentraciones del gen PAX5A, debido a que cuando no está el gen, la fluorescencia es muy baja, y cuando el gen está, la fluorescencia es muy alta. Y we have this author called Jankin in the 2012, and she said that when the primer is extended by the polymerase and form a double strand with the HP, a complete nuclease cleaver site will be formed. And that is an affirmation that it's true. Finally, we have the conclusions. Science has been in charge of demonstrating that the molecular biology is the pillar of the early detection of the disease and their subsequent prevention, focusing to the medicine of the future to a differentiated diagnosis for each patient to improve their health, and that it's necessary to understand the main causes of the diseases as common as B lymphocytic leukemia that we work in this investigation to provide timely treatment and exceptional care to our future patients, to be able to have several points of view apart from conventional treatment that we used to use in the medicine nowadays. Thank you.